I just want to give you a little check on the foundational materials that you've received, that you really have these concepts honed in. Firstly, on the concept of what extremophiles are and what they're not. So extremophiles, they require extreme conditions for optimal growth. And you also get extremotolerant organisms. So you should be able to differentiate between the two. There are those that actually need the extreme conditions, and then there are those which can survive in the extreme conditions but don't necessarily require them. And so when you look at this, it was because biology was constrained by anthropocentric assumptions about life. That means that we're not looking at life just as it presents itself. We're looking at it through the human lens. So this was largely based on laboratory grown organisms and human compatible conditions. So when you see anthropo, it means us, right? So this is why extremophiles were difficult to really comprehend, because we couldn't even think of these organisms that were able to live in conditions that are so foreign to us. So uh, this bias delayed the recognition of life's true physiological range. Now, when extremophiles were discovered, that gave us this huge paradigm shift, because now we had to ask the real questions about how these organisms were actually doing. So the discovery of thermophilic microorganisms in hot springs fundamentally changed the prevailing conditions of biological dogma. It demonstrates a couple of things. One, that protein stability is not universally limited to moderate temperatures, so there's more going on there than we thought. Cellular membranes can also remain functional far beyond what we previously assumed. So if you think about what you learned about the fluid mosaic model of how membranes work, you are told that there's a limit at which the membranes basically disintegrate because they get too liquid, right? But clearly, there's more to this than meets the eye, because you've got hyperthermophiles that live in very hot water or hot conditions. Then the third thing that was fundamental was life's boundaries are shaped by molecular adaptations, not fixed rules. So these are the take-home messages that I really want you to get. If you look at all the materials that you've got so far to lay your foundation about studying extremophiles, what we're really, as we start to understand how these adaptations are made, we're looking at the real functions of proteins and how they're able to be stable at these temperatures, because that's the molecule that causes all the life reactions to be able to happen, enzymes and all the rest. Also, cellular membranes. I want you to revisit the fluid mosaic model for how membranes actually are, and start think about um, how that can be adapted in order to survive much higher temperatures. And also think very hard that about life's boundaries, rather than them being anthropocentric, meaning from our perspective what is comfortable for us. They're shaped by molecular adaptation. So it's really about what the molecules can do under these conditions. So think about this. Why did early microbiology fail to detect extremophiles despite their abundance? I want you to be able to come up with one or two reasons why you think that is. And these are simple definitions that you should be able to provide. The difference between an extremophile and an extremotolerant organisms. You should be able to explain why you think extremophiles were historically overlooked and they're discovered so much later. And how did their discovery change the way we viewed biology? So this is what I want you to focus on as you're studying the foundational materials about extremophiles. Now, we've already introduced the different classes of extremophiles, the ones which are to do with the environments where they exist. So each category reflects a specific physical chemical constraint that is acting on the actual biomolecules. So when we first of all described all these different extremophiles, we're actually talking about where they're found, what condition. But what I want you to think of as we press on is the physical chemical constraints that actually acting on the biomolecules in terms of all these pressures that are there. Also, the concept of polyextremophily, meaning that you have more than one type of extremophilic trait. So you need to think about this. Why is it there? And why is it, why are polyextremophiles so much more common in nature? Which stresses often co-occur in natural environments? So I wanted to revisit the information you have with these questions in mind. And of course, you should be able to define all the different types of extremophiles. Then on the distribution across the trees of life, because we talked about the tree of life, the archaea, the bacteria, the eukarya. And now I want you to be thinking more in terms of what kind of examples of extremophiles are found in each domain. So as you think of these different extremophiles across the domains of life, you should think in terms of the archaea, the bacteria, the eukarya, and don't forget the fungal examples as well. And you should actually be able to name specific species that are found in each of these domains. So one of the peculiar things about archaea is that they possess unique molecular features. And these are the things that I would love you to start thinking about. Like if you look at the archaea as a group, you can start with this, that they have these ether-linked membrane lipids. Remember I said that you should be looking at the membranes of these organisms and seeing how it is that they're able to withstand those temperatures, especially for thermophiles. The same is true for psychrophiles. Each of those physical conditions, they're going to challenge the biology in different ways. So you can start with thinking about the archaea and how this unique molecular feature, which is that they've got ether-linked membrane lipids, how does that actually confer stability? So that's your little deep dive for yourself there. Now coming to the question of evolution. So one thing that is really key is this concept that they're ancestral models, not biological outliers. So most people are drawn to extremophiles because they seem so different. They seem like biological outliers. But in biology, we treat them more as ancestral models because they tell us more about early life. So where early life may have originated, because those were probably under extreme conditions. Modern moderate environments may represent derived niches, not original ones. Extremophilia is not an anomaly, but a deep evolutionary trait. So this is why extremophiles are so important, because they're able, not just on the biotechnological level, where they give us genes that you won't find in other organisms, but they can tell us the history. They can tell us far back into the history of life on Earth. They can give us a chance to analyze that. So this is why extremophiles are so important to astrobiology, because they give us this ability to look back into evolutionary time, to see about the biology that the oldest biology that we have and how it's developed from there to what we see today. So this is why we say this perspective reframes extremophiles as ancestral models, not as biological outliers. So I'd love for you to shift your thinking of extremophiles not as these strange, anomalous creatures that you find in these odd places, but more as organisms that enable us to look at the ancestry of life on Earth. So here you should discuss whether extremophily should be considered as ancestral or derived. And I've provided you with some deep dive material which looks deeper into this concept. So after you've read the deep dive material, you should be able to have this discussion with your colleagues and see whether you see it as ancestral or derived. So check your understanding to understand which domain dominates in extreme environments, and are extremophiles restricted to archaea? You should be able to explain whether the answer is yes or no. So moving on from that, they also have a role in ecosystems. So as we'll be looking at later, we'll see how it is that they drive biogeochemical cycles. how they cycle these nutrients. They support food webs in environments where there's no sunlight. And an environment like that is like the black smokers in the hydrothermal vents that we talked about before, deep at the ocean floor. In such environments, there is no sunlight to drive photosynthesis and drive a normal food web. So, how do those organisms survive? So, extremophiles have this unique ability to actually have very important ecosystem roles. And they're also very influential in the global elemental fluxes of these different nutrients. They demonstrate that ecosystems can function independently of photosynthesis. So this flies in the face of what we really learn for most of the time in biology. We think that all food webs depend on some kind of a primary producer, which is a photosynthetic organism. But extremophiles have shown us that that's not the case. Life can actually function independently of photosynthesis. So this is why extremophiles are also really interesting to study how ecosystems can exist in very bizarre places where you don't have the normal energy source that you'd expect. So this brings us to why we use extremophiles to study life on other planets or in other like interstellar bodies and things like that. Simply because if you're having something like the moon Europa, which is covered in meters and meters of ice, but underneath, at its core, it's got a warm uh, core that's still able to give off energy. So if you look at a situation like that, it's actually possible for life to exist under all of that ice, even though there's no sunlight that reaches the water under the ice of that moon. And so you can see how bodies in the solar system or beyond that people thought were completely lifeless could actually have life if they have a source of energy like what we see here. So if here on Earth, in the hydrothermal vents, you can see that there's actually whole ecosystems which are independent of sunshine. That means even in the solar system, places that we thought were completely barren could possibly be having life deep down, underground, under ice sheets, as long as there's some kind of source of energy. So this is why extremophiles are also really interesting because they tell us things like that. They give us new ways of thinking about how life survives. So, in terms of evolutionary and biotechnological implications, we study extremophiles because they inform early life conditions on Earth. They also tell us the limits of cellular evolution and also help us to discover new genes, new enzymes that enable life to actually happen in all these bizarre conditions. These insights directly motivate metagenomic exploration of extreme environments. So if you're asking why is it that we look at extremophiles, why are people like me fascinated by them? Why are they important to huge pharmaceutical companies around the world? Because they directly motivate uh, this metagenomic exploration of these extreme environments. This is why we study these extremophiles. This is why we look in extreme environments to look for new types of life, because there's new genes, new enzymes, new answers about how life was on early Earth, new answers about the limits of what a cell can actually do. And all these questions are what people are asking today. So this is why we study extremophiles. This is why they're key. And you should understand these reasons for why we're doing what we're doing in this course. So check your understanding. Can you name an ecological role of extremophiles in in nature? Can you think of what they actually do, like a specific one? Why are extremophiles important to evolutionary theory? So that is a few questions you can ask yourself after reading all the material you've gotten so far. And I hope this particular video clarifies the important things that you should come away with after reading everything that you've received, watching all the videos that you've seen. All right, thanks. I'll see you in the next one. Wait a minute. Actually, I have some questions. I would love to for you to comment and say if you've ever come across an extremophile before. Have you ever met an extremophile before in your life? So think about that. and think about the environments where you've been. And don't forget that you can also have uh, anthropogenic places, that is, man made places where extremophiles live. And you can have natural places where extremophiles live. So I'd love for you to say where you think you've actually come in contact with an extremophile and speculate what that extremophile could have been. And then speculate even further to think about what could have been a biotechnological use for the extremophile that you have encountered. Put that in the comments, and I'll pick some of the interesting ones to put in the next video. And we'll discuss how it is we actually look for the genes once we found an extremophile. How do we use metagenomics? How do we use classic genomics? All right, I'll see you in the next one.